SnapTag and ClipTag, an innovative protein labeling technology that can be used for the detection and study of protein function in vitro and in vivo in living and fixed cells. SnapTag and ClipTag can be used in a number of applications, including the labeling of proteins in cells, the study of biochemical cellular assays, live animal imaging, protein pull-downs, protein immobilization onto a variety of surfaces, and detection of proteins in gels. The following tutorial explains the mechanistic basics of this technology. A number of vectors containing the SnapTag or the ClipTag are commercially available. The sequence coding for the protein of interest can be cloned into these vectors using standard cloning techniques, such as traditional cloning using restriction enzymes, or assembling the protein sequence with the vector sequence containing the tag using any builder. The gene sequence coding for the protein of interest can be cloned on either side of the tag. Expression of the SNAP or ClipTagged construct will result in the synthesis of the protein of interest, in blue, attached to the SNAP or ClipTag, in yellow, as a fusion protein. The next step is to label the expressed fusion protein. For this, in the case of a SnapTag fusion protein, a SnapTag-specific substrate is used. This substrate consists of two parts. The first part is a benzyl guanine that is recognized by the SnapTag. The second part is a functional group that can be used to detect the protein, for example, in the case of a fluorophore, or to attach the fusion protein to another biochemical molecule or surface. Benzyl cytosine-based substrates that are recognized by the ClipTag are also available. The use of both tags and their specific substrates allows for dual labeling applications. The most common tool used for the detection of proteins in vivo or in fixed cells is fluorescent labels. Permeable and impermeable fluorescent labels that emit at different wavelengths are available. This characteristic provides flexibility to the system, as different wavelength fluorophores can be used for the labeling of the same fusion protein in different experiments, or in the same experiment at different time points, for example, in pulse chase experiments. During the labeling reaction, the functionalized benzyl substrate is attached covalently to the SnapTag or ClipTag. This reaction results in the release of free guanine in the case of SnapTag or cytosine in the case of ClipTag. The linkage between the fusion protein and substrate is irreversible. Now the sample can be exposed to a light source that will excite the fluorophore of choice. The fluorophore will emit light at a specific wavelength, which can be detected with the appropriate instrumentation. Now that we have explained the basics of this technology, let's go ahead and look at a specific example. In this case, we will be looking at the detection of a fusion protein of tubulin and the SnapTag in living cells. Tubulin is the protein that polymerizes into long chains to form microtubules, one of the components of the cellular cytoskeleton. In this case, we have a plate of eukaryotic cells that have been transfected with a SNAP tubulin fusion construct and thus express the SNAP tubulin protein. These cells now contain tubulin units that are endogenous wild type in blue and tubulin units that have the SnapTag fused to tubulin, shown in yellow. When the SnapTag substrate, shown as green bubbles, is added, the fluorophore moiety will be covalently bound to the SNAP tubulin protein, releasing free guanine. The cells can then be exposed to a light source emitting light at the correct absorption wavelength, shown as a purple flash. The fluorophore attached to tubulin will emit a fluorescent signal at its determined wavelength, shown as a green fluorescent light. This will be captured by the microscope using the correct filters to detect that emitted light. Here is a picture of the microtubule arrays, shown in green, being detected using the SnapTag.